ever wonder how scientists compare DNA sequences to detect mutations, study evolution, or even identify conserved genes? Well, in bioinformatics, this is done using sequence alignment. And in this video, we'll explore global sequence alignment using the Needleman-Wunsch algorithm, focusing on how the scoring matrix is built and how traceback works. So here we have two sequences we're going to be aligning. The first sequence runs down the left side, and the second sequence runs across the top of the matrix. In global alignment, we compare both sequences from start to finish. Before filling the matrix, we define a scoring scheme. And in this example, a match scores +8, a mismatch scores -5, and a gap scores -3. These three values determine every number in the matrix. We begin by initializing the matrix. The top left cell is usually 0, representing two empty sequences. Moving right across the first row and down the first column, we repeatedly apply the gap penalty by adding it to every current cell to get the next cell. So our gap penalty is -3. We add it to the current cell, which is 0, to give us the next cell, which will then be -3. Then the next cell will be -6 after adding the gap penalty to the current cell. Now we reach the most important part of the algorithm, filling the matrix. Every internal cell in the matrix is calculated using the same rule, and understanding this rule is key. So for any given cell, we consider three possible ways of reaching it. First is a diagonal move, and this represents aligning two characters, one from each sequence. If the characters match, we add the match score. If they do not match, we add the mismatch penalty score. Second is the vertical move, and this represents introducing a gap in the horizontal sequence, so we add the gap penalty score. The third is the horizontal move, and this represents introducing a gap in the vertical sequence, again adding the gap penalty score. After calculating all these three values, we choose the maximum, and that becomes the value of the cell. Let's calculate one cell step by step. So here we are aligning character C or letter C from the vertical sequence with G from the horizontal sequence. Since they are different, this is a mismatch, and the diagonal score is the top left cell, which is 0, plus the mismatch penalty score, which is -5, and the result is -5. Note, you can write it in your rough sheet so you won't forget when it's time to compare. The vertical score is the cell above, which is -3, plus the gap penalty score, which is also -3, and the result is -6. The horizontal score is the cell to the left, which is -3, plus the gap penalty score, which is also -3, and the result is -6. We then go ahead to compare these three values and select the highest one, which in this case is -5, so we write it into the cell. The next cell is computed in a similar way, where we align C from the vertical sequence with A from the horizontal sequence. Since they are also different, this is a mismatch. And the diagonal score is the top left cell, which is -3, plus the mismatch penalty score, which is -5, and the result is -8. The vertical score is the cell above, which is -6, plus the gap penalty score, which is -3, and the result is -9. The horizontal score is the cell to the left, which is now -5, plus the gap penalty score, which is -3, and the result is -8. We then compare these three values and select the highest one, which in this case is -8, so we write it into the cell. This exact same process is repeated for every remaining cell in the matrix, moving row by row from top to bottom, while applying the same three-move rule each time. After filling all the cells, the matrix is now complete. At this stage, the numbers represent the best possible alignment scores for every prefix combination of the two sequences. The highlighted cells show the path that leads to the optimal global alignment score. So once the matrix is filled, we perform traceback to recover the actual alignment. Traceback always begins at the bottom right cell, which contains the final global alignment score. and in this example, it's 27. So at each step, we ask, which move, diagonal, vertical, or horizontal, produced this value? A diagonal move means the two characters are either matching or mismatching. So, for instance, we ask the question, how did we arrive at 27? The two letters in the vertical and horizontal sequence is a mismatch, and that tells us that the diagonal score, which is 32, was added to the mismatch score, which is -5, and that gave us 27. A vertical move introduces a gap in the horizontal sequence, and a horizontal move introduces a gap in the vertical sequence. So we follow this path backward, cell by cell, until we reach the top left corner of the matrix. This traceback path represents the sequence of decisions that produced the optimal alignment. After completing traceback, we obtain the final alignment, meaning we write out every letter or character involved in the traceback and the three rule score that led to the values representing them. Our final score is 27, which confirms the value in the bottom right cell, and this is the optimal global alignment score under our scoring scheme. This score reflects the best possible end-to-end alignment of the two sequences. In recap, global alignment compares sequences end to end. The matrix is filled using three possible moves per cell. Traceback converts the matrix into a meaningful biological alignment. If you understand matrix filling and traceback, you understand global sequence alignment.